Hello folks, this is David Villar and welcome to this first video on toxicants that cause convulsions. We're going to break this module on many different videos because there are many things that target the central nervous system. On this first slide, things that cause convulsions usually go along with other clinical signs which can help us make a differential diagnosis. Now, among all the different substances that we have on this slide, which are just a few of the most common ones, we're going to try to cover most of them. In here we have strychnine, pyrethroids, organochlorine insecticides, OPs, carbamates, sodium toxicosis, 1080, pennitrem, zinc phosphide, methylxanthines, amphetamines, cocaine, bromethylene, and metaldehyde. Now you can find a good reference in Ivy's, which is called the Beasley's Notes. He used to be a professor at the University of Illinois. It's a bit old, but it's the classic source and probably the most comprehensive collection of all the toxicants that are important in veterinary medicine. On this first presentation, we will discuss strychnine, tetanus, and metaldehyde, and we're going to basically follow the list in Beasley's Notes. There are also so many things that are impossible to cover, and I will simply mention them in white on each presentation. So starting with the strychnine, it's a bitter alkaloid extracted from the seeds of an Indian tree known as Strychnos nox vomica. It has long been used as a pesticide to control or kill gophers, rats, coyotes, and any other animals considered as pests. It's often dyed in red or green colors, as we see here. All the species are susceptible, but most problems are going to be in dogs that, as you know, are not very picky eaters. And many times there is malicious intention behind intoxications. On this slide we have an approximate lethal dose for different animal species. We also see the molecular structure with two nitrogen molecules that make it an alkaloid, which is important because when the animal dies, the toxin will be in its higher concentration in the stomach. And that is actually the place where we have to look first in a dead animal, because it does strychnine does not cause vomiting either, compared with other convulsant toxins. Now, with regards to the mechanism of action, it works as an antagonist of the neurotransmitter glycine at the postsynaptic neurons in the spinal cord. This results in unchecked reflex stimulation with predominance of more powerful extensor muscles, which cause rigidity. And as you probably all know, glycine is an inhibitory neurotransmitter which hyperpolarizes the neurons, and that decreases the likelihood of depolarization or number of action potentials going through the spinal cord. Now, the purpose of those reso cells or inhibitory neurons is actually to prevent an excess number of action potentials in the spinal motor neurons. Now, with regards to its toxicokinetics, it's poorly absorbed in the stomach because it's ionized in an acid environment. Once it gets absorbed in the intestines, it becomes metabolized in the liver and is excreted with the urine. And when the animal dies, the highest concentrations are going to be present in the stomach contents. What are some of the features of an intoxicated animal? Well, as we mentioned, there is a predominance of extensor muscles over flexor ones. So the dog is going to show stiffness and rigidity in all four limbs. That rigidity also inhibits respiration, resulting in hypoxia and asphyxia. Any external stimulus will trigger spontaneous violent convulsions and opisthotonus. And some features that are relevant compared with other convulsive agents is that it does not cause vomiting, neither paddling, nor chewing, nor salivation, or running fits like we saw with sodium fluoracetate. The treatment involves preventing asphyxia and convulsions. We can use pentobarbital to effect and artificial respirations to reverse those two conditions. As a muscle relaxant, we can use methocarbamol This is usually unnecessary if we use pentobarbital. If we do not have pentobarbital, we can use gas anesthetics, and for decontamination we can use an emetic if we are early before the onset of signs, and if we are past that point, maybe we can go with activated charcoal and a saline or osmotic cathartic, which is also effective to bind the strychnine in the GI tract. And finally, fluids and forced diuresis will probably speed up the elimination through the urine. Now let's move on to the next one, tetanus. The toxins are produced by Clostridium tetani, which is a gram-positive spore-forming bacillus that grows under anaerobic conditions. Those spores are very ubiquitous in the environment. They are present in the feces of many animals, and once they get into the soil, they can persist there for many years. Now, poisoning is known to result from clostridial growth in a contaminated wounds, which have closed and there is anaerobic conditions. It's possible for the original point of entry to heal without any evidence of the infection, and this is actually the most common portal of entry in horses, basically a deep puncture wound. In foals, umbilical infections also occur, particularly if they don't receive maternal immunity. In cattle, the usual portal of entry is via the reproductive tract at parturition. Castration and dehorning with elastic bands can also result in clostridial infections. And in pigs, castration wounds are the most common point of infection. And in lambs and adult sheep, infection is most often associated with either castration, docking, or shearing. Actually, docking with an elastic band is probably the most common cause of tetanus in lambs. Now, with regards to the mechanism of action, the main exotoxin, which is known as tetanus spasmin, which is actually a huge protein of about 150,000 Dalton of molecular weight. Now, this toxin is going to travel in a retrograde transport through the nerves and to a lesser extent via the bloodstream until it basically reaches the brain stem and the spinal cord. Once it gets into the spinal cord, it's going to block the action of inhibitory neurons by preventing the release of glycine and GABA in their presynaptic membranes. And this basically prevents all the inhibitory control from those interneurons, and any sensory stimulus will trigger the excitation or activation of motor neurons, which is actually going to result in continuous muscle hyperactivity and rigidity. And death usually comes as a result of rigidity of the respiratory muscles and the associated asphyxia. On this slide, we see which species are most sensitive compared with the guinea pig, which is the standard species used for comparisons. The most sensitive by far and away are horses, which are even twice more sensitive than guinea pigs. As we can see, dogs and cats are several hundred times more resistant compared with horses, and birds are thousands of times more resistant. So it would be almost impossible to see cases in birds. Now, the predominant clinical signs are characterized by sustained tonic contractions of muscles without actually any twitching. Here we see a dog with a sawhorse stance. There is a protrusion of the third eyelid and a contracted facial expression, which is called a sardonic grin. There is also extension of the tail, which has been described as a pump handle tail, and you can tell there is a persistent rigid extension in all four limbs in the animal. Now you can pause this slide to read in more detail the clinical signs and their description. It includes tetany, sawhorse stance, hyperesthesia. We can also see convulsions. And basically, and also there could be dysphagia, particularly in horses, which is caused by paralysis of the laryngeal and pharyngeal muscles, and that could actually end up in aspiration pneumonia. With regards to the treatment, we could try to find the offending wound and meticulously debride it. If we can get a hold of the antitoxin and give it IV, it will reduce the uptake of the toxin by the nerves. However, the antitoxin will not actually penetrate within the CNS, so it will not really reverse the clinical signs. We can concurrently give the vaccine or the toxoid because the natural infection does not induce immunity. Antibiotics will decrease bacterial numbers. And finally, muscle relaxants will obviously help to counteract the rigidity in those extensor muscles. Now, if the animal is recumbent, the prognosis is generally poor, about a 50% chance of survival in horses that are displaying neurological signs. With the last one is going to be metaldehyde. This is used as a molluscicide, that is slug and snail baits. As you can see, the structure is a polymer of 3 acetaldehyde molecules, and most incidents will happen in dogs because of their nosy nature. If we look at the minimum lethal dose, it's about 100 milligrams per kilogram, which would be considered a moderately toxic compound on an acute basis. There is not much difference in the LD50 between species. We have the dog, cat, rat, mouse, rabbit here, very similar in most of them. And the way it works is by liberating acetaldehyde by gastric hydrolysis in the stomach. It smells actually like formaldehyde, and it seems like it decreases the concentrations of 2 neurotransmitters, serotonin and norepinephrine in the brain. And that appears, and as a result of that, we see signs of hypersalivation, vomiting, convulsions. Those convulsions seem to be more continuous than with strychnine poisoning. And also any external stimuli does not seem to have a marked effect as seen with strychnine toxicosis. Now, something which is also a telltale sign is severe hyperthermia up to 107 degrees, which has been called as a shake and bake syndrome. Death usually occurs from respiratory failure, and it takes place within a few hours. And if the animal survives this period, it may succumb to liver failure within 2 to 3 days. Now, early decontamination before clinical signs develop would be ideal. And once the animal presents, there is really no specific treatment. Everything we can do is really provide symptomatic and supportive care. Methocarbamol for tremors, anticonvulsant agents like diazepam for agitation and convulsions. For severe cases, we may need gas anesthesia and artificial respiration. Forced diuresis will not really speed up the elimination because very little goes into the urine, but it will help to control dehydration, acidosis, electrolyte imbalances, and because of the acidosis that usually goes along with metaldehyde, sodium bicarb would also be indicated in most cases. So this is the final slide. On the next video we will talk about other common convulsive toxicants. So until next time, thanks for watching, and I'll see you on the next one. Bye bye for now.